Okay, so this video is going to talk about gene regulation, and we're going to use the example of lactase production in bacteria. This has been a really historically important system for learning about how regulation works, and I think it's also a really great way to introduce ourselves to concepts and also build skills for analyzing these kinds of systems. So here's what we'll be going through in this video. We're going to be emphasizing why there's regulation, what sorts of functions are fulfilled by having regulation. We'll compare lactase regulation in humans and bacteria, and then we'll take a deep dive on the players in lactase regulation in bacteria, learning how to analyze mutations in various of these players, and learning how to use regulatory diagrams to analyze this kind of situation. Okay, to start out, let's remind ourselves what we learned about lactase regulation in humans and in most mammals. So remember, in most mammals and in most humans, there's a system where in childhood, lactase is produced and OCT1 is not produced. So lactase is produced and this gene OCT1 is not on. It does not produce OCT1 protein. And then we switch over to adulthood, in which case now OCT1 is produced. Those OCT1 proteins come down and bind to a site somewhat near the lactase gene and shut off lactase production. Okay, so in bacteria, the best one that's been studied is E. coli. Bacteria can also live on lactase. It can lactose. It can digest lactose. And it has a lactase-like protein called beta-galactosidase. And it's actually related to lactase. And so I've decided we're just going to call it lactase for the purposes of this video. And so similarly, just like us, where most mammals only drink milk for part of their lives and therefore only want to produce lactase for part of their lives, E. coli similarly can digest lactase but only wants to do it at certain times. So the first time it wants to do it is when lactase is present, and the second part of that is when there's no better option. So when glucose is present, E. coli doesn't need to digest lactase because actually glucose is a better energy source. And we can kind of clearly see why lactose is an inferior energy source, because we can see right down here that glucose is produced from lactose. So if you already have the glucose, then why bother to do the extra step of digesting lactose to make more glucose? Okay, so we can start out with our system of the OCT1 repressor, and we'll kind of start by looking at some of the similar pieces in the bacterial system, the bacterial lactase production system. So in this case, instead of OCT1, we're going to call the protein that represses lactase, we're just going to call it the repressor. And instead of having this sequence that we talked about that OCT1 binds to, there's a different sequence that the repressor binds to in E. coli. And we're going to call that sequence the repressor binding site, or the RBS. And so we're also going to change the shape of these things for a reason that will be useful in a second. So that our system now looks like this. So similarly, we have a protein produced by a gene that binds near lactase and turns off lactase. Another difference is that whereas in the case of mammals, the OCT1 gene is sometimes on, it's on in adulthood and not on in childhood. In this case, it's different. In this case, the repressor protein is on pretty much all the time. So already we can see that the logic's going to have to be a little bit different here. So let's see how this works. Okay, so we said that the repressor protein can bind to the RBS and turn off lactase. And so that's what we see in the absence of lactose, that if there's no lactose around, then the repressor is going to bind to the RBS and turn off lactase. Okay, well, what about in the presence of lactose? Well, now lactose is going to come and bind to the repressor, and it's going to cause the protein to change shape. The repressor is going to go through a conformational change, which affects the binding site. And so now what happens is this repressor that's bound to lactose cannot bind to the RBS, and so now cannot repress lactase, and lactase will be on. And so then we can kind of work through how this works. So in the absence of lactose on the left, we have a repressor with this kind of open site for binding the RBS. But in the presence of lactose, the repressor undergoes a conformational change. So on the left, in the absence of lactose, it binds the RBS. In the right, it does not bind RBS. So on the left, it represses lactase, and on the right, it does not repress lactase. And so we have both the sort of the total outcome of the circuit, that lactase is repressed when there's no lactose and not repressed when there is lactose. And we also have the kind of step-by-step logic of the circuit. Okay, so another way to think about this, these diagrams are great for building our intuition, but then once we have that, we want to work towards something somewhat more simplified to allow us to analyze and think through these problems without having to draw out all the proteins every time. Okay, so in this case, what we've talked about is lactase production on the right. That's the overall outcome, right? That's what we're interested in. Is lactase being produced or is it not? So what we learned is that the closest thing to lactase production, the thing that's the proximal cause that's turning lactase production on and off, is binding of the repressor. And does the binding of the repressor turn it on or off? Well, it turns it off. So we can mark this with this block diagram here in red. So that block means that if the repressor is bound, then lactase production is the opposite. So if yes, then no. But if no, if the repressor is not bound, then yes. So you see that when you see one of these block diagrams, that it's going to be the opposite. If yes, then no. If no, then yes. Okay, well then what? Now we can take it a step further. What affects repressor binding to RBS? Well, what affects the repressor binding is whether or not lactose is bound to the repressor, remember. And which way does that work? Well, when lactose binds the repressor, it stops the repressor from binding. So if this is a little bit fast, just go back and look at the notes or watch the last bits of the video so you can get a sense that when the lactose binds the repressor, then that stops the binding to the RBS. And so again, we're going to use one of these block diagrams showing that if yes, then no, if no, then yes. And so now we can go through the logic. If the lactose binds to the repressor, then that makes the repressor not bind to the RBS, and that leads the gene to produce lactase. If instead the lactose does not bind to the repressor, well, now the repressor is in the conformation when it can bind to the RBS, and that leads to shut-off of the production of lactase. Okay, so we've now gone through the logic for how E. coli senses when lactose is present. And I just want to point it out that this part is kind of like in mammals. In mammals, during childhood, lactose is present, so we want it on. And in adulthood, lactose is not present, so we want it off. The details are different, but the logic, kind of the sensing logic, is the same. Okay, so the next thing we can do with what we've learned is to think about how different kinds of mutations are going to affect lactase production. And so now we have three parts. We have the repressor gene, we have the repressor binding site, and we have the lactase gene. So let's go through it step by step. So the first thing is we can imagine an R- mutation. So that would be repressor minus. We mean here a mutation that knocks out repressor function. So this means no repressor protein is produced. So whether or not there's lactose, we're not going to have repressors. And so that means whether or not we have lactose, we have no repressor, so we have no repressor binding to the RBS, and so we're going to have production of lactase. And that's the case whether we have lactose or whether we don't have lactose. And so we can start to build up a list here. So we said that in the case of wild type, that is with no mutants, The first case we went through, that when lactose is present, that we're going to produce lactase, and when lactose is not present, then we're not going to produce lactase. And now we've said that if we don't have a repressor protein, then what's going to happen is whether or not there is or is not lactose, that we're going to produce lactase. The cell will produce lactase whether or not there's lactose present. Okay, let's think about another kind of mutation. So the other thing we can imagine is an RBS mutation, a mutation that knocks out this repressor binding site. So remember, this is like how the OCT binding site is knocked out in people who have lactase persistence, that they do not have a binding site for OCT1. Okay, so now let's go through the logic of what happens. So now in the presence of lactose, the repressor undergoes a conformational change when it binds to lactose, and so cannot bind to the binding site and produces lactase, just like in the wild type, in the normal. What about in the absence of lactose? So remember, usually now what would happen is the repressor would bind, but now, though the repressor is in the conformation to bind, there's no site for it to bind to, so again, it cannot bind, and again, lactase will be produced. Sorry, there should be mRNAs here as well. There they are. Okay, so now we can add this to our list. So we said that we analyzed a repressor minus mutant, and now we have our repressor binding site minus mutant. So there's actually another interesting kind of mutant, which is a protein sequence change in the repressor gene. So in a nucleotide change that leads to a different protein that's created. I've noted that here with this little nub on top. And what this does is this keeps the repressor from being sensitive to lactose. So now, unlike the wild type repressor, the lactose cannot bind to this so-called super repressor. And what that means is whether or not there's lactose, it can bind to the RBS. So now in the absence of lactose, just like in the wild type, the repressor will bind and it will shut off lactase production. However, now even in the presence of lactose, the repressor cannot bind to lactose, and it still therefore is going to bind to the RBS and repress lactase production. Okay, so now we have a different kind of mutant. The first two mutants we looked at were going to always produce lactase, but this mutant, this super repressor, is never going to produce lactase, because whether or not there's lactose, it's going to bind to the RBS. Okay, so we've kind of finished up with the when lactose is present, the system for how to sense lactose in E. coli. And so now let's talk about the second part of this, which is that the cell has a way of sensing whether there's a better energy source, that is sensing whether or not there's glucose. Okay, so to remind us, in the case of lactose, we have on the left, if there's no lactose, we have the repressor in the binding conformation. It binds the RBS and represses lactase. On the right, when lactose is present, it changes the conformation of the repressor by binding to it, does not bind the RBS, and does not repress lactase. So we have the details, but we also just have this logic of no lactose, repression of lactase, lactose, no repression of lactase. So what about for glucose? So for glucose, the overall output is the opposite, right? In this case, if there is glucose, then the cell does not need lactase, so it wants to repress lactase. And if there is no glucose, then optimally, the cell will not repress lactase. Turn lactase on and digest the lactose if it's present. Okay, so let's talk about how this works. So similarly to the system with the RBS, in this case we have a different protein which is called CAP. And CAP is going to bind to a CAP binding site, CBS. So let's go through how this works. All right, so if CAP binds to the CBS, I've just gotten rid of the RBS because we're not going to be talking about it now for a bit. If the CAP does bind to the CBS, now that's going to turn lactase on. Okay, so just note that this is different than the case with the RBS. Here, binding of CAP is necessary for lactase to be produced, instead of binding being repressive for lactase being produced. Okay, so that's the first little difference that we have. And so that difference here, notice that the CAP binding, this is a positive regulatory site, whereas repressive binding is a negative regulatory site. Binding here stimulates or activates lactase production, but binding here represses lactase production. So that's one way in which the system is different, plus versus minus on these binding sites. So let's take a step back and just kind of go through. So we now know, before we get into the details of CAP binding, we now know we have two different sites, right? We can have CAP binding and then we can have repressor binding. And so we've said that if CAP is binding, then that's good for lactase production. And if the repressor is binding, that's bad. And so that it stands to reason that if we have binding of CAP but not binding the repressor, that lactase will be produced. However, some of these other ones, it's not clear, right? So in this case, we have binding of CAP that's going to tend to activate lactase, but binding of the repressor is going to tend to not activate it. So here we have one positive signal plus one negative signal, and our question is lactase going to be produced? So it turns out to be the case that no. If the repressor is binding, then lactase will not be produced whether or not CAP is bound. Okay, what about the other way around? So now we do not have binding in the repressor, which should favor production of lactase, but we also don't have binding of CAP, which should disfavor production of lactase. So what's going to happen under these circumstances? So again, we have mixed signals, and again, this is going to be no. So in general, of these four possibilities, lactase is going to only be produced when CAP is binding and when the repressor is not binding. If you think about it, this makes sense. So remember, over here we're sensing whether or not there's glucose, and here we're sensing whether or not there's lactose. So we only want to produce, the cell optimally would only produce lactase if there is lactose, which is to say that there's no binding here, and if there is not glucose, which we will learn is when this is binding. So we only want to do it when both of these signals are saying, yes, go ahead. There's no glucose and there is lactose. So do produce lactase. So this will become a little clearer as we go along, but I first just wanted to point out the way that these two signals get integrated into production or non-production of lactase. Okay, so now let's get into the details of how glucose sensing works. So as with the case with the repressor, so this CAP protein has two different forms. On the left, we said there are basically two forms. On the left, we have a form that does not bind the CBS, and on the right, we have a form that does bind the CBS. And the difference is binding of this other molecule called cyclic AMP. Okay, so you can already see things are getting a little bit, so it's not quite as straightforward as lactose being bound to the repressor. Here there's an additional intermediate molecule. So this is going to take a little bit more of a pathway to work our way down. So let's go ahead and dive in. So cyclic AMP is this molecule right here, and it is mostly produced by an enzyme called adenylyl cyclase, which converts ATP into cyclic AMP. So it turns out that in the presence of glucose, that this enzyme is blocked. And so what that means is that we do not produce cyclic AMP. So when there is glucose, there is not cyclic AMP. And when there is not much glucose, there is more cyclic AMP. Okay, so let's work this out step by step. If there is no glucose, we just said that there will be cyclic AMP. That cyclic AMP is then going to bind to the CAP proteins, and the CAP proteins are going to undergo a conformational change that allows them to bind to the CBS, and which then stimulates lactase production. It has to be bind for lactase to be produced. Okay? What about the other case? Okay, if we do have glucose, then we do not have cyclic AMP. The CAP proteins do not bind cyclic AMP, and they stay in this closed formation so that they do not bind to the CBS, and lactase is repressed. Lactase is not activated. And so this gives us our general output that when there is glucose, we will not produce lactase, and when there's no glucose, we will produce lactase. Okay, I know that it's a couple more steps, right, than we were before, and we will be getting to this with our diagram, so sit tight, we'll be reinforcing this. Okay, so in fact, let's go ahead and reinforce it. So here's the whole pathway. When there's no glucose, we do have cyclic AMP, and when there is glucose, sorry about that misspelling, we do not have cyclic AMP. Cyclic AMP either binds or does not bind to CAP, which either leads to binding to the CAP binding site or not binding to the CAP binding site, which either activates lactase or does not activate lactase. And so here are the details: glucose to no glucose, CMP, CAP change, binding to the CBS, activating lactase, and then the opposite: glucose, no CMP, no binding to CAP, no binding to the CBS, do not activate lactase. And so we've got both the details, and then we've got the general logic, or the general outcome. If we have glucose, we do not activate lactase. Sorry, if we do not have glucose, we activate lactase. If we do not have glucose, we do not activate lactase. Okay, so we can build this into our regulatory diagram. So on the top, we have our lactose sensing pathway that we already talked about. And now we can build up our glucose sensing pathway. Okay, so the step that's closest to lactase production is binding of the CAP protein to the CBS. If it binds, does that stimulate or block lactase production? Well, if you'll remember, binding of the CAP protein stimulates or activates lactase production. So instead of putting a red block, we're going to put a green arrow. So that means positive. So if this is bound, if this is yes, then this is yes. If this is no, then this is no. Okay, so the next step, what determines whether the CAP protein binds to the CBS, is whether cyclic AMP binds to CAP. And again, this is a positive arrow. When cyclic AMP binds to CAP, that stimulates CAP binding to CBS, which stimulates lactase production. Okay, so now we've got two steps in our regulatory diagram on the bottom here. And then our final is glucose. And again, when glucose is present, this is going to block the production of cyclic AMP, and therefore block cyclic AMP binding to CAP, and on down the line. Okay, so now we have our whole diagram for the glucose sensing pathway. And so now we can work through what the logic looks like. Okay, so if there's no glucose, well, this is a block, so that means it's backwards. If this is no, this is yes. So yes, we'll have cyclic AMP binding to CAP. This is forward, so if yes, then yes. And again, same thing, if this is yes, then yes. So now we can go all the way, just from this diagram, we can go all the way from glucose to lactase production just by flipping things from no to yes. And again, if glucose is yes, then cyclic AMP binding to CAP is no, CAP binding to CBS is no, and the lactase will not be produced. And so together now we have we have the top and the bottom, the lactose and glucose sensing pathways that together are controlling lactase production. Okay, so just as we did with the repressor system, let's go through some different mutations involved in glucose sensing. So the first one would be if we have a mutation in the CBS that prohibits CAP from binding. So what do we expect here? Okay, so let's go through the two possibilities. If there's no glucose, then we have cyclic AMP. Cyclic AMP binds to the CAP, but because there's no binding site, it cannot bind near lactase, and so therefore it does not stimulate lactase, and lactase stays off. Okay, the other case would be if we do have glucose. So now if we do have glucose, we have no cyclic AMP. The cap stays in the non-binding form and now definitely will not bind. There's nothing to bind and there's nothing to bind to. And so again, lactase will be off. And so we can add this to our list of mutants. So down here we have if the CBS site is, or if the CBS is mutated, is negative, then we will not produce lactase either in the presence or in the absence of lactose. Sorry about that, I apparently duplicated my slides. Okay, so let's do another one. So another one would be, what if we have a mutation in the CAP gene such that we never produce CAP protein? So I'm going to use this stippled thing to mean there's a mutation in the gene. The reason there is before I was using red X's either to mean a mutation of a gene making it non-functional, or meaning repression of a gene making it not on at that moment. So I'm going to change that, and we're going to use this stippled thing to mean a mutation that inactivates this gene. So if we have no cap, then that means we can't have binding, and it doesn't matter whether we do or do not have glucose, right? So if we do have glucose, well, we don't have cyclic AMP, but it doesn't matter because we don't have any cap protein, so we'll have no binding, so lactase will be off. If we do not have glucose, now we do have cyclic AMP, but still we don't have cap protein, so we have no binding at the CBS, and so again lactase will be off. So if some of this stuff is going a little bit fast in the video, what I would suggest is just to pause it and go through the reasoning by yourself in hopes that you can check back and forth, go back 20 seconds and see my explanation and see if it makes sense. Rather than just letting me ram forward, make sure you get it bit by bit, because this is how we're building up our skill set to analyze these things. Okay, and so now we have another possible mutation here, a mutation in the CAP protein, the gene encoding the CAP protein, so that we don't produce CAP protein. And now we neither produce lactase in the presence of lactose or in the absence of lactose. Okay, so now here's an interesting one, and this actually exists. This is observed in different strains of E. coli, which is the super binding cap. Okay, so now instead of a normal cap, we have a mutation in the cap gene that produces a cap protein with a different conformation. And you can see this conformation has the open binding site even in the absence of cyclic AMP. So it's going to bind whether or not there's cyclic AMP. Okay, so this is different. Just like the cap minus, the situation is not sensitive to cyclic AMP, but now we, instead of never having binding, now we always have binding. And so we expect lactase to always be on. So let's go through this. If we do not have glucose, then we expect to have cyclic AMP. And it might bind to the cap, might not, but in either case, the cap will bind and stimulate lactase. So that's just like the normal wild type situation, right? But in this case, so now if we do have glucose, now we don't have cyclic AMP, it does not bind to cap, but cap can still bind because it's a super binder. And it can still stimulate lactase and lead to lactase production. And so now this one is different. So here we have this mutation, the cap-SB, which is going to produce lactase whether lactose is or is not present. Okay, so we have one more piece to talk about here. So we've talked about how the repressor, so now we're back to thinking about lactase sensing. So we talked about how the repressor binds to the RBS and controls whether lactase is produced or not. And we said it shuts off lactase when it binds, and when lactase binds to the repressor, lactose binds to the repressor, that stimulates lactase production. And so the key thing here is whether lactose is present in the cell. And we've just so far said, like, well, we've just asked, is lactose present or is it not? So there's one more step here. If lactose is present in the environment, in the medium that the bacteria is in, that doesn't necessarily mean that lactose can get into the cell. Lactose does not just diffuse through the cell wall, through the cell membrane. In fact, it needs a little bit of help. So what that means is that even if lactase is in the environment, if it can't get into the cell, then the repressor is still going to stay in the repressing conformation. It's going to bind to the repressor binding site and turn off lactase. So in fact, for lactose to get into the cell, it needs one more thing, which is another protein. It's a protein called permease. And the permease basically is just a channel that allows the lactose to get into the cell. And so if we have permease, what that means is now the lactose can get into the cell, and now we have the familiar situation where the lactose binds to the repressor, changes its conformation, causes the repressor not to bind, and lactase to be produced. Okay, so permease is itself a protein, so that means it's encoded by another gene. Okay, so now we have the lactase gene, we have the CAP gene, we have the repressor gene, and now we have the permease gene, all of them producing different proteins. So what happens here is the permease is on, it's on pretty much all the time, and that leads to production of permease protein, which goes to the cell surface and lets the lactose in. And so actually we can add this to our regulatory diagram. Should this go on the top or should this go on the bottom? Well, the permease is allowing the cell to sense lactose, right? So it's kind of on the top part of this pathway. And we can just add that before lactose can bind to the repressor, the permease needs to allow the lactose into the cell. And so this is controlling that, and it's positive, right? If the permease allows lactose in, that allows the lactose to bind. If the permease does not let it in, that does not allow the lactose to bind. And so we can just extend our lactose sensing pathway by one step. Okay, so now let's think about a mutation. So again, this stippled effect means that now let's imagine that we have a mutation so that we do not have a functional permease gene. We do not have a gene that can produce a functional permease. So let's think about what's going to happen. In the absence of lactose, in the environment, there's no lactose in the cell, so the repressor is going to bind and it's going to turn off lactase. In the presence of lactose in the environment, it still cannot get into the cell because we don't have a functional permease, and so the repressor is still going to bind. So despite that there's lactose available, it's not actually available to the cell, right? Because it can't get into the cell where it's needed. It's needed either to be digested by lactase or to block binding of the repressor. And so whether or not we put these cells in the presence or absence of lactose, lactase is going to be repressed because the lactose cannot get into the cell. And so we have our permease mutant down here, our permease minus, and we see that whether or not there is lactose, we do not expect production of lactase. Okay, so those are all the components we're going to talk about today. Those are the major components of lactase regulation in E. coli. And I just want to point out we haven't talked about a few other things. And that's kind of for two reasons. So one reason is that when this system has been studied, mutations in these other important parts have not been isolated, right? So you can imagine it's important for glucose to get into the cell, and if glucose can't get into the cell, then we'd also have a problem, or it would change the behavior of the system. But those aren't observed, and so we haven't talked about them. Similarly, for any of these genes to be expressed, we need an RNA polymerase, and we need ribosomes to transcribe and translate the genes respectively. And we haven't talked about those, even though they would have an important effect. The reason, certainly for these other two, that we don't observe these is probably because a mutation in the RNA polymerase, if we had an RNA polymerase that didn't function, well, that wouldn't just affect lactase production and lactose digestion. It would affect everything, right? It would affect all the genes, all the machines and proteins that need to be made in the cell. And the same thing with the ribosomes. And maybe the same thing with the glucose transporter, or it may be the case that glucose can simply diffuse into the cell and does not need a specific permease-like protein to get it in. So I just wanted to point that out in case people were wondering why we were obsessing about these particular few things. It's basically because those are the ones that were observed in the laboratory while studying E. coli, so those are the ones we know about. And one more thing is it sort of relates back to our case of OCT1 in humans. So you might have noticed that when we said that lactase persistence is always due to a mutation near the lactase gene. But potentially you could also get a lactase persistence state by having a mutant OCT1 that cannot bind lactase. And probably, again, the reason for this is whereas lactase kind of only does one thing, the lactase gene produces lactase. The OCT1 protein, the OCT1 gene and the OCT1 protein, binds to many different genes. It regulates many different genes. We've talked about how it regulates lactase, but it actually regulates many different genes. And so the problem with getting a mutation in OCT1 is it could give rise to lactase persistence, but it would probably have a lot of other impacts. So it would be so costly, it would have there would be so many other effects, so many other disease-like effects on humans that we don't see this OCT1 mutation. We only see the mutation near the lactase gene. Okay, so I just wanted to talk about one more thing before we summarize. So here I've made some choices in terms of naming. And I've named all the things we're talking about in terms of function. Historically, the names were different, and part of that is because historically, when this system was being studied, they didn't know what the different functions of the genes were or the different components. They were just finding that mutations in different kinds of different kinds. And so the lactase gene wasn't called lactase, it was called lacZ. The repressor gene wasn't called the repressor, it was called lacI. The RBS was called lacO, and the permease was called lacY. So this means that if you look at somebody else's problems on this, you're just going to have to kind of translate from biology 355 speak into this other system, so that, for instance, what we call repressor minus, they will call I minus. And then the final thing is just to remember that lactase is actually, what we've been calling lactase, is actually a related enzyme, which is called beta-galactosidase. So let's just summarize what we've gone through. The main point here is this is the beginning. This is a really nice first system for us to talk about the general characteristics and system of gene regulation. This is stuff that matters all the way from humans to bacteria and all the way from lactose sensing to developmental biology to toxin regulation, all sorts of things. And so one thing to point out here is we saw two kinds of regulatory relationships where the repressor represses lactase transcription and production, and CAP, where binding activates lactase production. Another concept we introduced was this concept of protein conformational changes. So when the repressor is bound by lactose, we said that causes a conformational change. Similarly, when CAP is bound by cyclic AMP, that causes a conformational change in these proteins. But remember that we also saw that it can be in either direction. So here, repressor binding lactose leads to no binding of the repressor to the binding site. And down here we have CAP binding to cyclic AMP. That actually stimulates the binding of CAP to the CAP binding site near lactase. So both at the top and the bottom here, we have a general theme, which is that we have regulation. So up here we have regulation of genes by binding of proteins. And here we have changes of protein conformations by binding of small molecules. But in each case, it can go in either direction. Here binding represses, here binding activates. Here the conformational change of binding stimulates, and it stimulates binding, and here it blocks binding. The other thing that we did was we went through mutations, and we talked about a variety of what their impacts would be. And what we found was that in pretty much every case, in every case actually, these mutations would lead to lactase either always being on or always being off. Basically, it led to a decrease in regulation, right? That these mutations knocked out the regulation and led to lactase being less regulated. And the final thing is we learned how to build and analyze regulatory pathways. So thanks very much. I hope this was useful. I look forward to seeing you in class.